Welcome. This video answers a question that sounds almost too basic to ask. Why can't you just run a t-test on genomics data? The t-test is the first test almost everyone learns. It is simple, it is fast, and here it is wrong in 6 separate ways. Over the next half hour we will take each assumption apart, see exactly how genomics violates it, and then rebuild the test into something that works. And here is the twist worth waiting for. The method most people end up using is still a t-test underneath. It is just a t-test that has been repaired. Let's start with the question that motivates everything. You have single nucleus RNA sequencing from the human cortex, say from the Allen brain cell atlas. You pick a gene, GAD1, which marks inhibitory neurons. You have 2 groups of donors and you want to know whether GAD1 expression differs between them. This is the most ordinary question in genomics. And the instinct, trained into all of us, is to reach for a t-test. 2 groups, 1 continuous measurement, compare the means. That instinct is what we are going to examine. Before we break it, let's be precise about what the t-test actually does, because the failures all follow from this structure. The numerator is the signal, how far apart the 2 group means are. The denominator is the noise, the standard error, which is your estimate of how much that difference would bounce around by chance alone. The ratio asks a single question. Is the gap I observed large compared to the gap I would expect from randomness? Everything hinges on the denominator being a trustworthy estimate of chance. Hold on to that. It is where the deepest failure lies. For that ratio to mean anything, 6 things have to be true. The measurement is continuous. The values are roughly normal within each group. The 2 groups have similar variance. Every observation is independent. The sample size is large enough to estimate the variance. And you are asking 1 question, not many. 6 assumptions. Genomics data violates every single one. Not sometimes. Structurally, by construction, all 6. That is what the rest of this video is. 6 failures in order. Failure 1. You are not running 1 test. Here is the arithmetic, and it is brutal in its simplicity. A p-value of 0.05 means that when nothing is going on, you will call a result significant 5% of the time. That is the deal you signed. Now run that deal 20,000 times, once per gene. 5% of 20,000 is 1,000. So even if not 1 single gene is truly different, you expect 1,000 significant genes. 1,000 genes with small p-values, ready to be put in a figure, ready to be enriched into pathways, ready to be believed. The t-test did nothing wrong here. It delivered exactly what it promised. The error was asking it 20,000 times and reading each answer as if it were the only one. The fix is multiple testing correction, and there are 2 philosophies. Bonferroni controls the family-wise error rate, the probability of even 1 false positive across the entire list. It divides your alpha by the number of tests, which at 20,000 genes means a threshold of 0.000025. It is correct, and it is so conservative that in a typical experiment you find almost nothing. Benjamini-Hochberg controls the false discovery rate instead, the expected proportion of your significant list that is wrong. You accept that 5% of your hits are junk in exchange for actually having hits. This is what genomics uses, essentially universally. But note carefully what 𝗿𝘂 gives you. FDR is a property of a list, not of any individual gene. There is no such thing as a gene with an FDR. Your gene does not have a 5% chance of being wrong. Your list has 5% junk in it, somewhere. That distinction gets lost constantly, and it matters. Failure 2. 3 samples cannot estimate a variance. This is the failure that does the most damage, and the one people think about least. Look again at where the standard deviation sits. It is in the denominator, which means if S is too small, t explodes. Now, with 3 samples per group, your estimate of S is desperately unstable. It has 2 degrees of freedom. It bounces around enormously from gene to gene, purely by chance. So somewhere in your 20,000 genes, some gene will have 3 replicates that happen to land almost on top of each other. Not because that gene is well behaved. Because of luck. Its S is tiny, its T is enormous, its p-value is beautiful, and it goes straight to the top of your ranked list. You have not found biology. You have found a gene that got lucky with its variance estimate. And here is the cruel part. This gene is not just a false positive. It outranks the true positives. Your most confident finding is your least trustworthy one. Here is that effect drawn out. On the horizontal axis is the standard deviation a gene happened to get. On the vertical axis is the t-statistic it produces for a fixed, identical true difference. The curve is a hyperbola. As s falls towards 0, t runs away to infinity. Notice this is not a gentle relationship. Between a gene with an s of 1 and a gene with an s of 1/4, the t-statistic quadruples, with no change in the underlying biology whatsoever. The signal was the same. Only the luck of the variance estimate changed. This is why raw t-statistics from small n genomics experiments are close to meaningless, as a ranking. The fix is one of the genuinely elegant ideas in the field, and it is worth understanding rather than just invoking. Here it is. You have 3 samples for your gene, but you have 20,000 genes, all measured on the same platform in the same experiment, with the same sources of noise. Most genes with a similar expression level have a similar variance. So instead of trusting each gene's 3 lonely points, you borrow strength. You estimate a typical variance across all genes at that expression level, and you pull each gene's individual estimate toward it. That is empirical Bayes shrinkage. A gene whose S came out absurdly small gets pulled up toward the crowd, and its t-statistic collapses back to something honest. The gene that got lucky loses its advantage. This is exactly what lemma does with its moderated t-statistic. It is still a t-test. The numerator is unchanged. Only the denominator has been repaired, using information the single gene t-test threw away. Failure 3. Counts are not measurements. The t-test assumes your measurement is a continuous quantity with a variance that does not depend on its mean. RNA sequencing gives you neither. What you get is a count, the number of reads assigned to a gene. 0, 1, 2, 17. It is an integer, it is bounded below its 0, and its variance is tied directly to its mean. That last point is the fatal one. For a Poisson process, the variance equals the mean. Count more, scatter more, automatically. Real RNA sequencing is worse than Poisson, because on top of the sampling noise there is genuine biological variability between samples. That is overdispersion, and it is why the negative binomial exists. Now look at what this does to a t-test. A lowly expressed gene and a highly expressed gene do not have comparable noise. They cannot share a variance assumption. Equal variance is not slightly violated here. It is violated by design, and the violation is systematic across the entire expression range. It gets worse at the bottom of the range, which in single cell is most of the data. Take a gene with a mean of 2 reads. What do the values look like? 0, 1, 2, 3, 4. Discrete, bounded at 0 and skewed to the right. There is no version of that which is normal. The normal approximation needs enough counts for the discreteness to blur away, and a low-expressed gene never gets there. And single nucleus data lives here. Most entries in the matrix are 0. So the t-test's continuity assumption and its normality assumption both fail hardest exactly where most of your data sits. Not at the edge. At the center of mass. 2 fixes, and they take opposite routes to the same place. Route 1. Stop pretending the data is normal, and model the counts as what they are. There's 𝗖𝗂𝘀𝘁 and 𝗂𝗺. Both fit a negative binomial generalized linear model, which builds the mean variance relationship into the model rather than assuming it away. Route 2. Keep the normal machinery, but tell it which observations to trust. That is 𝗁. It measures the mean variance trend in your actual data, converts it into a precision weight for every single observation, and hands the weighted data to lemma. Low counts get downweighted because they are noisy. High counts get trusted. Both routes work. Both are used in production. And notice what route 2 really is. It is a t-test again, with weights that repair the variance assumption. Failure 4. And this one is about the escape route people reach for. So the data is not normal. The standard reflex is to reach for a non-parametric test. Drop the t-test, run a Wilcoxon rank sum. Problem solved. Except it is not, and here is the arithmetic that ends the idea. With 3 samples in each group, there are exactly 20 ways to arrange 6 ranks into 2 groups of 3. Under the null, each arrangement is equally likely. The most extreme possible outcome, where your 3 treated samples hold the 3 highest ranks, has a probability of 1 in 20, which is 0.051 and 0.102. So the smallest two-sided p-value a Wilcoxon test can physically produce, with n equals 3 per group, is 0.10, not 0.05, not 0.01, 0.10. Even if the separation is perfect, even if every treated value is astronomically higher than every control value, the test literally cannot reach significance because it has thrown away the magnitudes and kept only the ranks, and with 6 observations there is not enough information left in the ranks. Non-parametric is not assumption-free. It trades a distributional assumption for a loss of power, and at small n that trade is fatal. There is a second misconception worth clearing up while we are here, because it changes how you report your results. People say the Wilcoxon test compares medians. It does not, except under an assumption almost nobody checks. What it actually tests is stochastic dominance, the probability that a randomly drawn value from group 1 exceeds a randomly drawn value from group 2. Those are different questions. You can construct 2 distributions with identical medians that a Wilcoxon test separates with a tiny p-value, because the shapes differ. Only if you assume both groups share the same shape and differ purely by a shift does the Wilcoxon become a test of the median. So, when you report a Wilcoxon result, be careful what you claim you found. And this matters more than it sounds, because Wilcoxon is the default test in Scanby's rank genes groups. If you have ever run single-cell marker detection with default settings, this is the test you ran. Failure 5. And this is the big one. This is the failure that does the most damage in single-cell work, and it is the easiest to commit without noticing. You have 3 donors per group, but each donor gave you 10,000 cells. So you run the t-test across cells, and now n is 30,000 instead of 3. Look at what that does to the standard error. It has the square root of n in the denominator, so going from 3 to 30,000 shrinks your standard error by a factor of 100. Your t-statistics explode. Everything is significant. Every gene. And it feels wonderful, because the p-values are astronomical and the volcano plot is full. But think about what you actually measured. The variability between cells within a donor is not the variability between donors. You have measured the wrong noise, precisely and confidently, and then compared your biological signal against it. Your 3 donors are still 3 donors. Sequencing more cells from them does not give you more donors. It gives you a more precise measurement of those same 3 people. This is pseudo-replication, and it is everywhere in the published literature. The fix is pseudobulk, and it is almost insultingly simple for how much it repairs. For each donor, for each cell type, sum the counts across that donor's cells. 1 donor, 1 number, per gene. You have collapsed 10,000 cells down to a single value, and it feels like you are throwing away almost all your data. You are not. You are throwing away the variability that was never evidence about your question in the first place. What is left, 3 numbers per group, is the actual unit of replication in your experiment, the donor. Now n is 3, because n was always 3. The standard error reflects between donor variability, which is the thing your biological claim is about. And now you can run 𝗁𝗁 or 𝗁𝗂𝘀𝗁 on those pseudo-bulk values, and get an honest answer. If you have repeated measures, donors sampled at more than 1 time point, then you need a mixed model with a random intercept per donor, using something like 𝗁. But the principle is identical. The donor is the unit, not the cell. Failure 6, and it is short, because it sits upstream of everything else. This one is upstream of everything else. Sample A got 20 million reads, sample B got 10 million. Gene X has twice the counts in A. Did expression double, or did sequencing depth double? The raw counts cannot tell you, and a t-test on raw counts will happily report the library size difference as biology. Worse, sequencing is compositional. The reads are a fixed budget being divided up. If one gene captures a larger share, every other gene's share falls, with no change in its absolute abundance at all. That is not a measurement artifact you can ignore. It is the structure of the data. So you need normalization, and the methods differ in what they assume. Median of ratios in 𝗍𝗂𝘀𝗁 and 𝗃 in age are both assume most genes are not changing, and use that assumption to find the scaling factor. When that assumption holds, they work well. When it does not, for instance in a comparison where a large fraction of the transcriptome genuinely shifts, they will silently rescale your real signal away. But assumption is doing the work, and it deserves to be checked rather than inherited. Let's put the whole scorecard on one slide. 6 assumptions. 6 failures. 6 fixes. Continuity, broken by counts, fixed by a negative binomial GLM or Voon weights. Normality, broken by discreteness and skew, fixed the same way. Equal variance, broken by the mean variance relationship, fixed by modeling that relationship. Independence, broken by cells nested in donors, fixed by pseudo-bulk or a mixed model. Enough N, broken by 3 replicates, fixed by empirical Bayes shrinkage. 1 question, broken by 20,000 genes, fixed by false discovery rate control. Now look down the last column. Every one of them is a repair to the same test. Which brings us to the twist, and the real point of this whole video. After all 6 failures, what does the field actually run? Pseudo-bulk your cells to the donor level. Normalize for library size. Apply Voon weights to handle the mean variance relationship. Shrink the variance across genes with empirical Bayes. Compute a moderated t-statistic. Control the false discovery rate. Now look at step 5. It is a t-statistic. A difference in means divided by a standard error. The thing we spent this entire video taking apart is sitting right there in the middle of the recommended pipeline. Because the t-test was never the problem. The t-test is a perfectly good instrument for comparing 2 groups. What failed was applying it to data whose structure violated everything it needs, and reading the output as if nothing had gone wrong. Every fix in that list is a repair to one assumption. Put all 6 repairs in place, and the t-test works. That is what limit is. That is what the moderated t-statistic is. A t-test that someone took seriously enough to fix. So to be fair to the t-test, when is it actually fine? It is fine when your data is genuinely continuous rather than counts. Think protein abundance from mass spectrometry, or a module score, or an imaging readout. It is fine when your N is large enough that the variance estimate is stable, which in practice means more than about 10 per group. It is fine when your observations really are independent, 1 measurement per donor. And it is fine when you are testing 1 gene, chosen in advance, for a specific reason. That last one matters. If you are testing a single per-specified gene with a real hypothesis behind it, there is no multiplicity to correct and no ranked list to be fooled by. The t-test is not a bad test. It is a test with a domain of validity, and genomics is outside it. The mistake is never that someone used a t-test. The mistake is using it without asking whether the data satisfies what it needs. 5 things to carry out of this. 1. The t-test fails on genomics data in 6 separate ways, and they are structural, not incidental. 2. The denominator is where the damage is. With 3 samples, your variance estimate is mostly noise, and the genes that get lucky outrank the genes that are real. 3. Non-parametric is not a rescue. At 3 per group, a Wilcoxon test cannot produce a p-value below 0.10 no matter how clean the separation is. 4. Pseudoreplication is the most consequential error in single-cell analysis. Cells are not replicates of a donor, and sequencing more of them does not add information about your biological question. 5. Every fix is a repair to the same test, and the repaired version is what the field runs. Understanding why each repair exists is what separates running a pipeline from doing statistics. This is the first video in the Statistics for Genomics series. Next we look at parametric versus non-parametric properly, what you gain, what you pay, and why the choice is not about escaping assumptions. Full tutorials, books, and code are at 𝗿𝗂𝘀𝗁.com. If you want to work through this on your own data, reach out at 𝗿𝗂𝘀𝘁.com/contact. Check 𝗿𝗂𝘀𝘁.com to learn more.